All right, so we're gonna do muscles, and then we'll do tissue. Then we'll do nervous tissue. After that, we'll start the next subject. Okay, so muscles. This won't take too long. Muscles. There's three types of muscles. Okay, muscles. Overall, their functions. There's a lot of functions to them. When we get into the muscular system, we'll learn all about that. But in general, it controls movement of the body. You want to change and move the environment if you want to do that, or walk from here to there. It also moves blood around the cardiovascular system. We have blood vessels around. We have muscle around blood vessels. We have muscle around intestines to help push the food forward. We've got muscles around the uterus, the urinary bladder. Those are the areas that most people forget about, that we have muscles. When people think muscles, they're thinking muscles. But there's a lot of other muscles too. With the heart, I mean, that's a muscle in itself. Okay? So the three types is this. Skeletal muscle, which is the muscles that most people think about. It's connected to the skeletal system. Cardiac muscle, which is obvious the heart itself. And the smooth muscle is what students usually forget, because that's muscle around all these internal organs, around the blood vessels, the urinary bladder, the uterus, the digestive system, and so forth. Okay? All right, so let's talk about the first one, skeletal muscle. Skeletal muscle is muscle that's very long, and they have what we call striations. We're going to see these lines that kind of go like this. And we'll explain what that is later on, how the arrangement of the microfilaments are inside each one of those muscles. But they create these, what we call striations. The nuclei over here, it's multinucleated. There's a lot of contractile organelles that are in there called actin and myosin. They're made of proteins. We have to make a whole slew of them, so we're going to need a lot of nuclei to start making them. So that's one cell right here. That's one cell right here. That's one cell right here. And there's a lot of nuclei. They're usually on the periphery of the cells themselves. And they're over here. Okay? These muscles, these muscle fibers over here, they're very long, but they don't branch. Okay? We have complete control about this muscle, so we call it voluntary muscle. This is connected to our bones, and we want to make that muscle contract. We want to move the environment. We want to pick up things. So we have control about that. So skeletal muscle is striated. It's not branched. There's a lot of nuclei there, and it's voluntary. You could move it. You have control about that. And this connects to the bones. So it kind of looks like this, kind of like how I just drew it for you. And on a microscope slide, you'll see this. And you see those wonderful striations there. Okay? Cardiac muscle. Cardiac muscle is long too, but they are branched. And we'll talk about why they're branched when we get into anatomy and physiology. Okay? But it happens to, the branching actually makes the action potential, electricity be able to spread a lot more faster. Okay? Because we have an action potential or an electricity that kind of goes this way, and then it can go two routes in the same rate, at the same rate. Okay? We want to do that when it comes to the heart. So you'll learn more about that later on. They are also, even though they're branched, they also have these striations. Okay? And the cells, they have special connections, these gap junctions that are over here. These are much thicker, or more prominent. The spaces in between, or the connections in between each of the cardiac muscle cells. These lines have a special name. They're special discs, and they're called intercalated discs. The nuclei here are not, well, each one of these cells is not multinucleated. There's going to be one nucleus in each cell. Okay? So this is cardiac muscle. It's only found in the heart. We don't have control about making this contract, so it's involuntary. The muscle fibers are branched. They're not multinucleated. They are striated, and they have special connections in between the cells, and they're called intercalated discs. So it kind of looks like this, and we look at a microscope slide, it looks like this. You'll see the striations in here. You definitely see the branching going on. You also see these darker connections between the cardiac cells, cardiac muscle cells, and those are called intercalated discs. Okay? The last one is smooth muscle. A smooth muscle tends to be a little bit difficult to identify on the microscope slides. But let me show you what it looks like schematically. They're gonna be like well, they're called fusiform shapes, the cells. They're football shaped. They are not striated, and given them that name, a smooth muscle, meaning a smooth texture. It's not branched, and there's one nucleus in each one of the cells. This smooth muscle is the one that most students forget about, but this is the one that's just as important as the other two. This is the muscle that's located around blood vessels and the intestines and the esophagus to help move the food forward, to help push the blood or move the food, to cause blood pressure to go increase or decrease in the blood vessels, be it around the uterus, around the urinary bladders, and so forth. Okay? This is what it would look like schematically. What it looks like under the microscope is this. It's difficult to actually see those fusiform shaped cells, but I'll help you along on the practical exam by saying, I'll put this up there and ask you, what type of muscle is this? Now, you do it by process of elimination. You know there's no stripes there. You know there's no branching there. Does that make sense? So it's only got to be one other one. I might ask you, where would you find this type of muscle? So it's a two-step process. You have to figure out what muscle it is, but that's not the question. The question is asking, where would you find it? And you can say around blood vessels, around the uterus. Okay? Questions on that? So those are the three types of muscles: skeletal, smooth, and cardiac. Okay? So let's look at what they would actually look like on your slides. This is skeletal muscle. You can't tell anything from there, so you got to get closer. Even that you can't. The only thing you can tell from that view and that view is that it's not branched. They're all going in a straight line. But when you get up really close, you start appreciating that wonderful stripe area there, the striations. See the striations over here, those parallel lines that you see. So that's skeletal muscle. Can't be cardiac muscle because you don't see intercalated disc and you don't see it branched. Cardiac muscles here, you can see there's branching going on. But when you get up closer, you can see more branching here, and really close, you can see the branching. You can also see dark intercalated discs over here. If you could actually get up closer, you could actually see the striations there also. But the intercalated discs are really nicely depicted there. Okay? This is smooth muscle, 40x, can't see much. 100x, even hard. There's 400, and there's 1,000. All right? I would give you something, like I said, I would say, what type of muscle is this? Where would you find this muscle? Okay? But there's no striations there. I mean, that's at 1,000. If you saw 1,000 for skeletal muscle, you saw the striations. All right? That's smooth muscle at 1,000. That's skeletal muscle at 1,000. See the difference? Okay? Okay. Questions on muscle? See, that was pretty easy. No? Compared to connective tissue and the epithelial, it's a piece of cake. Alright? Alright, now, nervous tissue, there's only one slide. Hey, thank God, right? Knock off the hard stuff first, then we go into this. Okay. Nervous tissue. Specialized cells that are going to give you these electrical impulses down its, what we call the neuron. There's two types of cells that you're going to see in nervous tissue. One is the actual neurons, which is the nerve cell that's going to produce this electrical impulse. And the other part is what we call neuroglia. Neuroglial cells are supportive cells. The best way I could explain the difference between the two is think of taking a lamp over here, you have a wire and you want to plug it in. The light goes on. The electricity comes from the wall and passes on the metal wire in there. But we have something around that wire. That's insulation. The neuron is the actual metal wire that's conducting electricity. The neuroglia is that insulation around there that's going to support it. It is not going to conduct electricity. It's going to make sure that the electricity stays within there in that wire to go from one point to the other. It's going to help aid and support the and that, when I say electrical impulse, it's really called the action potential. You get to know that very well in a week or two. Okay? But that's what that is, electrical impulse. Okay? And that insulation that's around here is neuroglia. We have 6 different types of neuroglia. One of them goes around the wire. The other 5 do other things, and we'll get into those later on. But it's all neuroglia. They're all going to help support the nerve. Okay? Or the neuron itself. Well, we'll get back to it. Okay. So the 2 main types is the neuron, which is actually a nerve cell, and the neuroglia. Okay? These are the non-conductive cells. The neuron is the actual one that's going to cause electricity, this action potential. There's 6 different types. You don't have to worry about them at this point. You will later on, but you'll learn about each one of them. The one that's going to put that that insulation around the wire is going to be either the Schwann cell or the oligodendrocyte. If we just take some of that insulation off, which by the way, the Schwann cell and oligodendrocyte make that insulation. The insulation in the nervous system is called myelin. You'll see the word come up in a couple slides or so. That's myelin in there to keep the electricity in the wire itself. If we take some of that insulation off and we plug it in, some of that electricity is going to escape from the wire and never reach the lamp. And the lamp won't turn on, or will just turn on very weakly. We have a disease that actually will destroy some of that myelin, will destroy some of that insulation and cause those issues to happen. It's a disease that we don't understand why it attacks the myelin, but it does, and shreds it away. It produces a scar tissue around those areas, and that scar tissue, which we call sclerosis, okay, when the nerve itself gets sclerosed, it then presses and disrupts the electricity from going forward. And there's multiple areas of that sclerosis that's happening on that wire. Do you know what that disease is called? Multiple sclerosis, MS. Okay? Our bodies, for people who have multiple sclerosis, they produce these antibodies to fight against the myelin. Why? We don't know. They're called autoantibodies. And it's an autoimmune disease. And you'll get to learn a bunch of them throughout the rest of AEP 1 and 2. Lupus is an autoimmune disease. Eczema is an autoimmune disease. Rheumatoid arthritis. There's a bunch of them out there. All right? Lupus and Graves' disease. Yeah, you'll get to know them as we go along. So the neuron has 3 parts. A neuron is going to look like this star-like thing with an end like that. That's what a neuron looks like. Very pretty. Yes. We have over here this one part here that contains the nucleus. That's called a cell body. It has a few other names like a soma too. That's the part that contains the nucleus. Then we have these other branches that come off. These are called dendrites. And then there's going to be one long one, and that's called an axon. You should be able to sketch a neuron and understand its parts. What happens here, well, this is an axon, and then it terminates here, and that's called an axon terminal. These are the 3 major parts of a neuron, though. That's where it ends. It's called an axon terminal. And it usually will end at a muscle. It's called a neuromuscular junction, which you'll get to know later too. What happens here is the electrical impulse from somewhere, the action potential, is going to go into the dendrites towards the cell body. The cell body is going to receive those impulses, do whatever it needs to do, whatever that impulse is, and then it's going to make an action potential go down the axon. The myelin insulation, the myelin is going to be around the axon only. And when this action potential goes down here to the axon terminal, it's then going to tell the muscle to contract. it might not go to a muscle. It may, now it wouldn't do both of this. I'm just creating a new creature here just for your entertainment purposes. But the axon might not go to a muscle. Instead, the axon terminal may go to a dendrite of another neuron and tell that neuron what to do. So it's either going to tell a muscle what to do or another neuron what to do. Is that clear? Okay. So the myelin, as I said, insulates the axon. And it's produced by the Schwann cells, or what we call oligodendrocytes. When we have a degeneration of this myelin, that's multiple sclerosis. So the dendrites, that's what it does. Cell body, that's what it does. And axon, that's what it does. I kind of explained everything over there, but you could read at your viewing pleasure later. Okay? This is just showing you similar pictures of what I drew. How they go to other neurons over here. You can see the myelin going over here. Showing the same thing. There's the axon terminal right over here to a muscle or another dendrite of another cell. So what this looks like is this. Okay? It's very difficult. If I give you a picture like this, I wouldn't ask you which one's the axon. You can't tell. How do you know which one's the longest one here? Does that make sense? But you could look at that and there's nothing else that looks like that. You can say that's a neuron. Okay? They may even look like this. We'll talk about different types of neurons. So what your lab slides would look like, there's at 40, there's at 100, and there's at 400. You can see there's a cell body right here. You can't tell where the axon is. And that's a really close up at 1,000. Okay? Questions about nervous tissue? Okay? Alright, the last thing I just want to do, it's only a couple slides, is abnormal cell growth. Because when you're using these terms throughout A&P1 and 2, few of them, the first three are pretty easy to manage. Abnormal cell growth, atrophy. Atrophy is when we have a decrease in cell number and cell size. Hypertrophy. This is when we have an increase in cell size only. You don't make more muscle cells when you lift weights. You make your muscle cells get bigger. Hyperplasia. This is an increase in cell number only. Alright? Now here's the other two that it's not hard to understand, but there's more to it. Something called metaplasia. Metaplasia is where, let's say, a certain epithelium is at, but then it changes, it adapts to a chronic irritation that might be occurring at that spot. For instance, you have pseudostratified epithelium in the trachea, which is one layer. Now you have a smoker that loves to smoke and puts smoke down that trachea. Well, that smoke can get in that one layer of tissue, the pseudostratified epithelium, and start destroying and irritating stuff underneath it. So the pseudostratified epithelium gets smart and says, fine, you want to smoke? Well, I got to protect my underlying areas here. So it changes, it adapts into something we can recognize, but in the wrong position. What's the type of epithelium that is used for protection? Stratified squamous. You have a lot of layers there, as in your hands, your skin, your esophagus. Well, this pseudostratified epithelium decides to change into stratified squamous. So now we can take a biopsy of this trachea, and if we see stratified squamous epithelium, we could recognize what it is, but it's in the wrong place. It's a well-differentiated cell type. We could recognize that, but it's telling us that something's irritating it. Smoke, smog, something like that. An infection could be doing that too. But this doesn't lead to cancer. Okay? This doesn't lead to cancer. There's other things that'll lead to cancer. You see metaplasia, it tells you there's something going on there. Okay? Smoking does cause cancer. I don't want people to think that I'm like lying about that. But it's being caused by other things. Not smoke itself, but what's in the smoke is a different story. Does that make sense? Okay? So that's metaplasia, when we have a chronic irritation happening there. Then we have dysplasia. Dysplasia is that the cells, wherever they are in your body, turn into different cells that we cannot recognize. They turn into stars. That doesn't make any sense. I can't even recognize it. These are more of a concern. Not that metaplasia is not. It's telling us something's going on there. But this one can lead to cancer. So what's happening here is this. These are normal cells up there. Here's atrophy. You decrease in cell size and cell number. Here's hyperplasia, where you increase in cell number. Here's hypertrophy, where you increase in cell size. This is metaplasia. This is what normal cells would look like, but then they change into something else that we could recognize. It's a well-differentiated cell. It's just in the wrong place. This is dysplasia. The cells themselves turn into stuff we cannot recognize. That's of a concern. We got to see what's causing that. This is why we do pap smears. Pap smears is going to actually pick up dysplasia. If we catch it early, that's a good thing. You could get rid of it. You don't have to do surgery at that point. You can do a little bit of surgery, but you could freeze it. You could do other things. There's the basement membrane. If those atypical cells, these undifferentiated cells, cross the basement membrane, you got cancer. You want to catch it before it breaks the basement membrane. That's why it's important to get the pap smears done once a year, because you want to catch it early. Okay? Questions about histology? That's it. Okay? Now, that's a lot, I realize. Now, let me just tell you, what I'm going to do with you guys, I have, there's a video. You have review questions to do on this, okay? Now, since you're in my lecture and lab, the theory is going to be in lecture. The theory and pictures will be in lab, okay? So studying for it is, you're hitting it on two ends, okay? There's nothing different about it, but there's certain questions that are a little bit easier to ask in lecture than to ask in lab. It's still studying the same stuff. Yeah?
What's the difference between the lab quiz and the practice?
Just a shorter version. 20 questions as opposed to 50, okay? So there's a few things on here I'm going to email later today, okay? One thing is, there's a video that's on the website that you can get. All right, it's a 45-minute video or something like that. I have it here. Yeah, it's this one here. I'm not gonna show you 43 minutes. Not gonna happen.
Hello, welcome to Anatomy and Physiology at Glenelg Community College. I'm Dr. Brent Arsham. For this video, we're going to look at histology.
What I find with this, do this afterwards. He kind of doesn't go in the order that I kind of taught you. So he kind of goes sporadic. So once you understand it in my order, you understand each part, then go to the video. Spend 43, 45 minutes, and he's going to give you, it's not everything. I would say he does about 70% of them, not everything. So, and he goes pretty fast. He has a very monotone voice too, not like mine. So it might cure your insomnia. But watch this. It's pretty good when you get to that point where you think you have a handle on things. He also does towards the end a little bit of a practical exam and ask you questions.
It's dense. What type of connective tissue do we find here and here?
And then he pauses.
This one is dense, irregular connective tissue.
And then he'll tell you what it is. And that's the way you should be studying. Okay? I know right now you're saying WTF and you can't understand what those are. You will get to know them. Okay? Trust me, you'll be able to do it. A lot of students do very well on these, the histology section. Okay? But you got to do it in an organized manner.